Hi everybody, we're ready to get started with today's Ensembl webinar. We'll be talking about Ensembl tools. My name is Emily Perry. I'm the Ensembl Outreach Project Leader, which means that my job is to, in various different ways, teach people how to use Ensembl and different features of Ensembl, which partly means doing webinars like this, means doing live courses, means making some of the online training materials that you can see. So what we're going to talk about today is we'll have a quick introduction to Ensembl, and then we'll start looking at some of our tools. We'll look at the Variant Effect Predictor, we'll look at BLAT, and we'll look at BioMart, and then I'll refer to you where you can get some help and documentation on using some of these things. So we'll start with the introduction. Ensembl is a genome browser, and genome browsers are really important because they take sequencing data, which is something which is very vast and is increasing at an exponential rate, and they put it into a form that's easy to understand, that's easy to access. So you start with something like this, which in itself is utterly meaningless, and we make it mean something. And there are sort of three major genome browsers out there. There's ourselves, there's UCSC, and there's also NCBI Map Viewer. So we have quite a lot of data in Ensembl. We have what we call gene builds, where we plot gene data onto the genome for about 70 species. We have gene trees which compare all of the genes that we've plotted onto all these species. We have what we call a regulatory build, which incorporates data from places like ENCODE, Roadmap Epigenomics, and in future Blueprint, and the Variant Effect Predictor, which we'll be looking at today. We have ways to view your own data. We have BioMart, another thing we'll be looking at today, which allows data export. We also have programmatic access. We have APIs. We have a MySQL server, which allow you to, if you're into that kind of thing, to access it programmatically. And we are completely open source. We have, as I said, about 70 species. We cover vertebrate species, and we try and cover a wide range of taxonomic space. We also try and cover biologically important species. So we try and cover species of agricultural importance, of medical importance, all the model organisms, and sort of conservationally important species as well. We also have some, if you look over here, you can see some non-vertebrates, which are used as outgroups for our comparative genomics analysis. So tools allow you to access and export Ensembl data. They allow you to perform very specific tasks, and they allow you to work with your own data in combination with Ensembl data, and that's what we want to talk about today. So the first tool we're going to talk about is the VET. So I'll do another quick poll, if I can make this thing launch. Have you used the Ensembl VET before? And we'll see how many of you have worked with this. The VET uses your own variation data. So you might have done some kind of variant calling. Maybe you sequenced a single gene. Maybe you did something like whole genome variant calling. Maybe you have a list of variation IDs. And what you want to know is which genes do these variants fall within, and how do they affect the variant, the genes that they fall within. Are they going to change the coding sequence, or are they going to fall somewhere intronic? Might they affect splice sites, regulatory regions, various different features like that? And you can get these kind of data from the VET, including, and these data has sequence ontology consequent terms. This is a kind of standardized set of nomenclature for describing different kinds of sequence features, for example, variation data. So we use the variation sequence ontology terms here, and we do have a page which lists all of these different terms. We also get SIFT and Polyphen scores. Now these are score changes. These are scores which reflect the change in amino acid sequence. They tell you basically how bad that amino acid change is likely to be, how much it's likely to affect the protein function. And this is based partly on how well conserved the protein is, and partly on the chemical change in the amino acid. And they have, I believe, the Polyphen cutoffs have changed, but overall they're ever so slightly confusing because they both score out of 1, but for some reason they have opposite as to what's bad for your protein and what's perfectly fine for proteins. So they will give scores, and they also give predictions for particular cutoffs. And I believe I do need to update my Polyphen slide here. And these scores, you'll see the scores and you'll see the predictions in Ensembl and when you use the VEP. So the VEP you can use to get these variant consequences, and there's a web interface where you can point and click with your mouse, and there's also a Perl script which is standalone, which you can use just through your computer, which doesn't necessarily need to communicate with the internet. So these are all the species highlighted where we have variation data in Ensembl, but actually all of the species in Ensembl will work with the VEP, including everything that we have in our sister site, Ensembl Genomes, which has plants, fungi, protozoa, bacteria, and other metazoa. So you can use all of these with the Variant Effect Predictor. Some of you might be wondering, well, my species isn't in Ensembl, like this T-Rex. Can I use that with the VEP as well? So we have the option to set up a cache. So if you're using the script, you can set up an offline cache, which allows you to run a VEP query completely within your own system. There's nothing that goes over the internet. You're just communicating with this offline cache with all the data in it. We make pre-built caches for all the Ensembl species. You can download them and work them in that way. But if you've got a GTF file and a FASTA file for your species of interest, you can actually make a cache and run that even if it's not something in Ensembl. So yes, if you had a T-Rex genome and you wanted to find out different variants between your various T-Rexes, you could do that. So I'm going to do a bit of a hands-on here. I've got a set of 4 variants, and I want to find out which genes they hit and what effect they have on them. I'm going to do this as a live demo. If you download the slides, there are sort of descriptors, there are screenshots going through what I'm about to go through in the downloaded slides. So if you want to have a look at them later, you can do, showing exactly what I've done. But I'm going to do this live, so I will just escape from my presentation, and I will select this set of variants. So this is just in what we call Ensembl default format, which consists of the chromosome, the start coordinate, and the end coordinate, which in the case of SNPs and single base insertions and deletions are of course the same number. Then we have the allele change, the strand, and a name for this variant, which is an optional thing to put in. So I'm going to go to the Ensembl homepage, and I'm going to click on Variant Effect Predictor, which is shown here right in the middle. I'm going to launch the VEP, and it's giving me a new job. I've got a box where I can paste in the data. It's already selected human for me, but I could open this up and select my other species of interest. I can upload a file or provide a file URL if I wanted to. There are different formats that you can put in, and there are examples of each of those here. So if you click on these, you'll see the different kinds of file formats you can put in. There are also options, you might need to open these up, of sort of different features that you can get out. So one of the things it's going to do is it's going to find out if there are variants already at the location where I'm looking at. So it will identify novel variants if they don't already exist in the database and be able to get this kind of data for me. It will also tell me if they already exist. It's also going to get me the SIFT and Polyphen scores I mentioned earlier, and it will get me, I can filter the data at this early stage if I want to. I'm not going to at the moment because I'm just putting in 4 variants. So I'm just going to click on Run. And now it's giving me a little table showing me that I've got this job running through the VEP. It's queued at the moment. It's counting down. I can save this. So it's finished now. I can save it, or I can delete it, or I can share this if I want to. I'm just going to click on View Results. And now you can see I've got all of my data. So I've got a summary of my results. I can see that I've got 1 novel variant that didn't already exist in the database, and I've got 3 that were already known. It's processed all 4 of them. I've overlapped 2 genes and 7 transcripts and 1 regulatory feature. These pie charts summarize the data, and I can also, I've got this very long complicated table with my 4 different variants. So I have It's changed the order of them. So I have var2, var4, var3, and then var1. You can see the different consequences. Each variant's hitting multiple transcripts, and some are hitting multiple genes, which is why we've got lots of lines for each variant. You can see we've got these different consequences that you can hover over and check what they mean. I can see the IDs of the gene and the transcript and the gene symbol that I've hit. And if I scroll across, I can see I've got these scores, these SIFT and Polyphen scores that I mentioned, which tell us how bad the protein change is. These are only obviously for the ones where there is a protein change. And you can see when I hover over them, I get the description. So if you're colorblind and you can't see the red and green, if you hover over, you'll be able to get the damaging or the tolerated or whatever it might be. And I can also see that these are the IDs of the variants that I've hit, which are already in the database. So there is quite a lot of data in here. I'm not going to go over everything. I've got some allele frequencies as well. For the known variants, I've got where there is an allele frequency known for different populations, I've got that as well. So there is quite a lot of data in this table. I'm not going to go over everything, but you can see there is quite a lot you can look at. And I can download this data, and I can also plonk this data directly into BioMart, which is one of the things we're going to be looking at as well in this webinar. I can see I've got some things that have appeared in the chat box, so let's see if there's anything useful. So somebody says VEP is, oh good, Will's been helping out, so that's useful. So somebody says, what are those benign and damaged? Is it effect? So the benign, the damaging, that's the predictions, these SIFT and Polyphen predictions, which are sort of computer algorithms which are based on the conservation of the protein overall and that particular base, that particular residue, whether or not it's a particular chemical change in the residue. And it's not known. It's just a prediction based on these features as to whether that particular change is likely to affect the protein function. So it's a really nice sort of predictive thing there. So I'm going to go back into the presentation, and I'll skip over those slides, and we will talk a bit about BLAST and BLAT. And I shouldn't have gone back into my slides because the first thing I'm going to do is go straight to a poll. So where are my polls? So the next poll, I just want to know who's used Ensembl BLAST. So there's another question from Sophia about the VEP and the predictions, asking if they're in the curated database. So we actually carry out on every base, on every protein residue that's in our database, we do SIFT and Polyphen predictions in human, and so you actually they're pulled straight out of the database rather than calculated on the fly, which is quite a good way to do it because it means that it works much more quickly than if they were calculated on the fly. So we have them for all our known variants, and you can also pull them out through the VEP for any novel variants that you've found. So we've got lots of people using, about 2/3 of you have used BLAST in Ensembl, about 1/3 have used BLAST but not the Ensembl one, and about 10% of you have never used BLAST at all. So I'll just close that poll and we'll have a look at Ensembl BLAST. So if anybody's not familiar with BLAST, BLAST is the basic local alignment search tool, and BLAT is a BLAST-like alignment tool. They're very, very similar tools, hence BLAST-like for BLAT. The main difference is that BLAT is a bit more rough and ready. BLAT requires much greater sequence similarity to work, whereas BLAST can work with much more loose sequence similarity. So depending on what it is you're trying to search, if you're searching a sequence from a species that you're fairly sure comes from that species, then perhaps you want to use BLAT. But if you're searching for something that's maybe from a different species, or an unknown source, something like that, you might want to use BLAST instead. And you can use it to search against Ensembl. You can search against Ensembl proteins, you can search against the genomes, and you can also search against cDNA. So we're going to go back to a hands-on, and we're going to look at a yeast protein sequence, and we're going to see if we can identify it in human. So again, I'm just going to select this sequence, I'm going to copy it, and I'm going to go to BLAST, which you can see in the top blue bar of any Ensembl page. So again, we've got a table of tickets. I actually did this search the other day, which is why it's shown up here. So anything that you've done in the past week, I believe, will stay here, and then you can save it to an account as well if you want to keep it longer. So if I do new search, I will paste in my sequence. You'll see it immediately recognizes that what I've put in here is a protein sequence. So it reads that and it says, yep, that's got M's and L's and V's and things, which are obviously not nucleotide sequence. It knows it's looking for protein. I can decide what database I want to search against, whether I want to look against the genomic sequence, the DNA or the protein, but we'll leave it just as genomic sequence for now. And there are some configuration options that you can choose. So because I put in a protein sequence, it gives me tBLASTn as my search tool, but I can add some configuration options if I want to configure my BLAST search in a particular way. I'm just going to do it on normal search sensitivity, but again, these are things that you can adjust. So this search sensitivity is very sort of crude. You're saying, yes, what you think is the correct parameters for this, I'll go with it. Whereas the configuration options, if you know exactly what parameters you want to go for, then you might want to consider going into these options here. I'm going to click on Run. And so it's added this as queued. Rather than wait around for that to work, I'm just going to click on this one that I did the other day. I'll click View Results. You see that one's now running, so we can wait for that, or we can just go here, which is exactly the same sequence. So what we can see is we've got some details of our job. We can download the data, and we have a table. So we've got the genomic location of all our hits, and we've got the genes that they overlap. We've got the orientation of the sequence on the genome, and where in the query our sequence starts and ends. Got the length of the alignment. We've got scores for each of these, e-values, and percentage identity. And we can also see a genome distribution. We can see that our best hit is this one shown in this box here. So that might be the one that we want to go to. I can jump to the genomic location. So if I click on this location, it will jump me straight there, and it will show me my hit as a box. It should show me my hit as a box. Here it is. So it's yeast sequence. That was the name I gave it, and it's showing me that it's going across these exons of this particular gene SMC4. Given that I got this yeast sequence from SMC1, that sounds about right, because they are paralogs of each other. Well, SMC1 and SMC4 are paralogs. So I will go, somebody's put a message in the chat, so I'll have a look at that. So, is there a maximum limit for sequence in BLAST? Do you know, I'm not actually sure if we have a maximum size. I would have to check that. I believe your email address is on the webinar system, so I can look that up, and I will send you an email when I have found that out. Okay, let's go back, and we're going to talk about BioMart as our next thing, and we'll have another quick poll just to see if you've used, who is familiar with BioMart, and we'll go into the last part of our story. So BioMart is a data export tool. It requires absolutely no programming. It is point and click with your mouse. and you use it to generate quick tables such as Excel tables, as well as things like FASTA files of sequence. And it's a web interface that allows you to mine Ensembl data. So you can find BioMart in the top blue bar of Ensembl, and there's lots of things that you can do with it. So what you do is you start by defining a set of something with combinations of parameters, so a region, a list of IDs, a particular function, particular phenotypes. So you define this list and you say, right, for all of these genes, variants, regulatory features, I want to export this data, which could be IDs, it could be sequences, it could be homologs, it could be a bunch of other stuff. There are so many options available, I cannot possibly put them on a single slide. And it works by a system of 4 steps. So you start by choosing a data set, so you choose which kind of database you're looking for, whether that's the gene database, a variation database, or a regulation database. You then filter the database. You say, right, I've got, say, all the genes in the database. I want all of the genes that match my filters, and so you're narrowing it down to the ones that you're interested in. You're then going to get attributes. These are the things that you want to print into your table, all of the bits of information that you want to have about the genes that you filtered down to. And then lastly, you get these data, these things that you want to print, you get them in your table or as sequences. So filters, just to go over, filters are additive, so when you do more than one filter, you'll get everything that's within both of the filters. So we'll do a quick hands-on. We've got a list of 6 genes, and we're going to get their function via Go terms. I'm just using 6, but you can use a much longer list than this. So I'm just going to close that. I'll select my list, and I'll go back into Ensembl, and I'm going to select BioMart. So I'm going to choose my database. My first thing I'm going to pick is that I'm working with the genes. So I pick Ensembl genes, and they are human genes. So I'll pick human genes, and now you'll see it gives me filters and attributes in this side menu. So I can start to filter by filtering the data. So if I go into filters, I can open up gene, and I've got this option here, input external references ID list, which is what I've got. I've got a list of IDs, so I'm going to paste them in. Just need to get rid of that, and so I replace that with a comma. They can be comma separated, or they can be carriage return separated. I have to tell BioMart what kind of ID I put in. These are just HCNC symbols, the sort of standard the human gene naming committee. That's not actually what it stands for, even though it sounds like it. And so these are the sort of standard gene names, and that's all my filters. So now if I click on count, I can see that I've got 6 genes out of the database, which is what I would expect. If I go into attributes, I can now get the features that I want for them. So if I open up gene, you'll see I've already got the Ensembl gene ID and transcript ID. I'm going to pick associated gene name, which is going to give me back that standard gene name. And if I scroll down and open up external, I can get these Go terms. So I'll get the accession name and definition. Now if I just click on result, I'm going to get a table. It will take a moment to load. So it's just giving me a preview table of 10 rows. The reason for this is if you were to do a query that was 100,000 lines long, it would take a bit of time to load. If you then realized that you'd missed off a column that you really wanted, that would be really irritating. And so you'd have to go back and reload it. By only loading 10 at a time, you can check that you've got everything you need, and then you can say, okay, I want to view all. So I can click on all, and it will open up a new page for me that gives me all the data that I'm looking at, which, as I said, because it's a lot of data, this one isn't 100,000 rows, but it does take some time to load. While we're waiting for that, I'll just go back to this page, the original page. You'll see that you can export data as different kinds of files, so you can get it as HTML, as comma or tab separated values, or as an Excel file, and you can also get it so that it emails it to you. So if it's a really big query, you might want to get it to send as a compressed web file that emails you when it's ready. No, still not loaded with this big data set. I'll just show you the first few lines. So we've got our Ensembl gene and transcript IDs, we've got our standard gene name, which was our original input, and we've got these Go terms which describe the function of our protein product. And as you can see, we've got multiple transcripts per gene, and we've got multiple Go terms per transcript. So there's quite a lot of data in this table, and it's still not loading. I give you my word that it will at some point load a big table there, but I'm not going to wait for it because it's just a waste of all of our time. These are also links as well, so you can click on these and go to the gene pages in Ensembl. I'll just see if we've got another. We've got another question about BLAST and BLAT. We can use both BLAST and BLAT for all genome and protein sequences. So if you're using, I think protein sequences have to be BLAST, and there are particular BLAST tools which are suited to protein sequences and designed for protein sequences. BLAT and BLAST can be used for genome queries. So the different tools are, when you put in certain kinds of sequence and you select certain kinds of things. So when you put in a protein sequence and say you want to go against the protein database, it will only allow you to do the particular kind of search which is possible for that kind of thing. So it doesn't allow you to attempt to do something that won't actually work. So we'll just go back into the webinar, and I'll skip over my demo slides. And so the last thing I want to point out to you is help and documentation. Help and documentation is absolutely vital. So we have loads of help and docs available. I think a lot of you got here via the EBI Train Online interface, where we do have some online courses as well as some fantastic other EBI training courses. We've got loads of tutorials. We've got some flash animations on our YouTube, and also if you happen to be in China, on your Youku channel. We've got ones on BLAST, on the VET, and on BioMart, as well as on a number of other topics as well. If you're ever stuck with anything you're using in Ensembl, email us. We are helpdesk@ensembl.org. We also have some public mailing lists that you can subscribe to as well. The announced mailing list just tells you about anything new that's coming out, whereas Dev is a kind of forum where people discuss using Ensembl in a programmatic manner. You can follow us on social media, on Facebook, on Twitter, and we also have a blog. We have publications. If you use our data or use our tools, we do appreciate people citing us, just like everybody else. We are reliant on grants, and if people cite our papers, we get better grants. And this is the team who works on Ensembl. This was us at our retreat last year. This is again the whole team and our funders, who we're very grateful to for funding this project. And just to point out, there are more webinars coming if you're interested, part of the EBI webinar series. So in a couple of weeks' time, there's React Home, we've got Unichem and Kemble coming up. If you are interested in these at all, please sign up and tell your friends about them and find out about more of the EBI resources. So that's everything from me. I have another question. Oh, I just have a thanks in the chat box. That was very nice. Now is the time to ask any questions before I go. I can, if there's a sort of raise hand button, if anybody wants to speak, I can turn on your audio if you raise your hand. Okay, so we've got the question is the best way to get a foothold on the e-training. So if you go to the Train Online site, these webinars are a great sort of intro to some of the stuff that's available. Many of the resources, I'll just open up Train Online because that's easiest. If I go into the course list for Train Online, you can see a lot of the courses that are available, and there are many courses that are beginner courses, so you could have a go at them. We also have these quick tours, which are quite a nice way to learn. So these are really a point for seeing what kind of things are available in a particular resource that the EBI offers. So it's really worth having a go at some of the quick tools and seeing actually if this is the thing, if this is the tool for you. If you decide that it is the tool for you, then I would recommend trying some of the longer courses for them. But the quick tools are a really good way to sort of check if you're actually looking at the right resource at all. We have a question about how Ensembl BLAST is different to NCBI BLAST. So we are using the NCBI BLAST software, so the actual algorithm and the way it works is exactly the same. The main difference is what it's searching. Ensembl BLAST is searching ensemble sequences, NCBI BLAST is searching NCBI sequences. It really depends kind of which annotation and what sequences you prefer to search.